Okay, so I think we're ready to get started. And this is actually the first seminar for this academic year. And there are many more to come in the fall semester, and I'm starting to work on the spring semester. It's a very good year. And we have a very good start with a distinguished guest, Steve Goldstein. And I know Steve for many years because the first time we met was on what used to be called the Cardiovascular and Pulmonary Studies section at the NIH. Doesn't exist anymore. In any event, Steve since then has moved to Chicago, so he's a neighbor. He received his MD and Ph.D. both from Harvard Medical School, was at Yale for a while, and then joined the faculty at the University of Chicago, where he's the head of Pediatrics, and also a director of an Institute for Molecular Pediatric Science, which I think sounds very interesting to me. It probably has a lot to do with mutations and so on, and congenital heart disease. And Steve has done and made very important conceptual contributions in the field of ion channels and ion channels in the heart. His work on IKS and KCNQ1, KCNE1 stoichiometry is very well known. And Steve, we're happy to have you with us. By the way, the title of his talk is SUMO Emerges from the Nucleus to Regulate Membrane Excitability, and I envision it as a big sumo wrestler.
Well, it is a pleasure to be here. First of all, thank you, sir, for the invitation. It's been wonderful to see old friends, and for the first time in this environment, and then also to see my son, who's sitting in the front row, I'm here embarrassed, who is a sophomore undergraduate here. The science going on is very impressive. In fact, the science going on here is so impressive that I am going to protect myself today, and I'm not going to talk about the heart. So if those of you who want to leave, I will just turn my back. If you like me enough after one presentation, since I'm 5 hours away, I'll come and talk about the heart. But I wanted to talk about something that's been engaging our attention for a little while, and which is fun because there's some ongoing political controversy about it, and maybe we'll get to some of that, maybe we'll get to some of that question. So the first thing I wanted to go. The first thing I'm going to do is just soften you up, which none of you need, so I'll move through it relatively quickly. But ion channels being the most important thing in the world to study and the only thing worth spending your career on, I need to at least give an orientation to that so that I know we're all on the same page. I do talk to the undergraduates and the graduate students that way when I lecture. And I tell them that everyone else tells you that what they're studying is important, but I'm right. And then I want to tell you two stories about SUMO. One that's a couple years old and one that is now submitted. And in the context of that, maybe we can talk some about the controversy that's currently going on. So starting about channels. Channels are the most important thing in the world. They give us sight and sound and feelings and motion, and you all know why, because you either study the heart or the eye or the ear or the brain or the muscles or whatever. And as a result of all those important roles, they also cause disease, and whether it's primary because we inherit mutations or secondary because we give drugs that bang on the channels and make things go wrong, or it's tertiary because there's somewhere in the pathway of the physiology or pathophysiology that we're studying, they are a wonderful substrate. And I suppose that it's within the ballpark to say that something like 40% of the pharmaceuticals that we give our patients act on ion channels or associating proteins in the membrane. So, just so we're all starting thinking about the same beautiful entity, this is as close as I get to my own crystal structure. Ion channels are transmembrane water-filled holes. And they are so powerful because they really only have to do two things. They have to open and close at the right time, and they have to leak the right material across the membrane down the electrochemical gradient. As a result, you can see beautiful single-channel traces like this from a sodium cardiac sodium channel, which opens when it's supposed to and then inactivates when it's supposed to, so that you have more than one heartbeat. And when many of them come together, you see fluxes like this. Again, you all know this, so I'm really going to try and move fast. I apologize for the introduction. There is, of course, the symphony of ion channels that is the heart that we will not talk about today, because we're going to talk about the more simplistic organization of the brain. And we will get there. I stole that from this morning's presentation. And of course, what I will get to eventually is showing you some of the first work out of my lab that has to do with something that's real, which is primary hippocampal neurons. And channels in all of these systems have to talk to each other. They have to do the right thing. And all of this complexity scares me to death. And so what I do is try as best as I can to simplify everything that we're studying down to what I call biology for geeks. So that ion channels are as simple as this if you're trying to understand how they work. If you're trying to keep things simple, so that the total current is equal to the number of channels that goes through the single channel and the open probability. And the open probability is all of cell biology, phosphorylation, dephosphorylation, stimulation, and so forth. Unitary conductance, inherent to the beast, number of channels of cell biology. So one can divide efforts that way. Now, getting into the things that we're going to talk about today. I want to talk some about potassium leak current. So potassium leak currents have been known or invoked for many years as key to setting the resting membrane potential. And as there is more potassium flux, the resting membrane potential moves down towards EK. And what you have in this example up here is that the resting membrane potential goes more negative, so that that cell, that excitable cell, is not firing, and then as it goes more positive, it can fire. So for many years, the question was, what is the source of potassium leak? It could be any number of things. In fact, it still is any number of things. Anything that will let potassium leak across the membrane will contribute to basal background potassium flux. We're not talking about membrane damage here, although even there you can have ion specificity. We stumbled some years ago onto some proteins that actually mediate this. And those are the ones that I want to talk about today. They are specific mediators of potassium leak currents or background currents. Again, most of you will recognize this. Potassium channels are formed by four forming loops coming together on the central axis. This is a picture from McKinnon's lab in 98 on the KCSH potassium channel. And generally, the classical potassium channels are formed by 4 subunits, each with 1 key domain. Today, I want to focus at first on these subunits with 2 key domains. And the first one that I'm going to mention is TOC. Now, this was in the early days of the genome sequencing projects, meaning about 6 months ago. No, probably in the early 90s, 93, 94. And we were looking through the emerging genome database, and we saw a potential open reading frame for a potassium channel that had 2 forming loops. Now, I understand if you don't study potassium channels, the response to that is, who cares? But for those of us who have dedicated our lives to potassium channels, this was the most exciting thing since sliced bread, because it meant maybe there's a different way to build potassium channels. So this is, in fact, from my Cerevisiae Baker's piece. There were a few reasons that we were interested in studying it, but we cloned it out. It was the only one apparent in the database. We expressed it in benefit oocytes because that was the closest thing we had to yeast cells to study at the time, because we didn't know how to batch yeast cells, but that's something we've now done. And it was a very interesting channel. I won't talk about it much today. Except to mention a couple of general things because I love it so much. If you imagine that you've got an entire plate of cells here that are white, those are normal Saccharomyces cells. And outside here, these pink cells are excreting something that's killing the white cells. And when you purify what they're excreting and put it in a disc, it diffuses out the other way and it kills them in that direction. So that's what that target is about. And that is because, that is why your socks taste good and your beer tastes good and your bread tastes the way it does. Because baker's yeast has an RNA virus in it that excretes something called killer toxin. And killer toxin goes and kills any yeast cell that doesn't have the virus in it. And therefore, it's a way of claiming your domain. That killer toxin goes and opens this potassium channel and causes the cells to die. All of that is relevant only because in going through the database and looking for channels like this, we found others from mammalian species that were related to this, and we wanted to try and study. So it was a timely stumbled on this that we found examples in the database for humans. And when we cloned them, there were about 40 isolates apparent in C. elegans, and a few apparent in rat and mammal genes. And every time we put them in our standard expression system and looked for function, what we got was a big, fat raspberry. None of them would do anything. The beginning of the controversy that's in the literature has to do with the fact that there is a group in France that will remain unnamed until someone else names it, who took this isolate and published that it worked to express current in oocytes. And they called it TWIC, or to-be domain weakly rectifying potassium channel. Anybody who spent their life studying oocytes was fairly convinced that what they were showing is stuff that you see all the time. Those of us who couldn't get any function out of these were really upset. And so this is how we felt. I go back to the yeast for a second. There's a reason to distract you. In order to try and get function from these guys, we were interested in using an expression cloning strategy. And what we did is we took yeast cells, again, this is something you're familiar with, that can grow when they're in 100 millimolar potassium, but can't grow when they don't have supplemental potassium because all of their potassium transporters have been deleted. So they're fine, except they don't have their potassium transporters. In order to survive, they need high potassium or something that will let potassium move across the membrane. And we took a Drosophila library, put it in these yeast cells, and said, show us anything that will let you grow. And our positive control was a channel called a plant channel from Arabidopsis cat1. And in fact, what we pulled out, we were stupid enough to name Dork for Drosophila open rectifying potassium channel. It had the body plan of all those mammalian and C. elegans genes that we've been looking at. And it acted like a classical golden Hodgkin cat open rectifier. It acted like an open potassium selective hole, meaning that there was no time dependence. When you changed the voltage, potassium just went through immediately at the driving force that you increased it. And that if you had symmetric potassium across the membrane, this is what your IV curve looked like. And if you had physiological potassium, where you had high potassium inside and low outside, you saw this rectification, an open potassium selective hole. I was incredibly excited. This is the whole, the Hodgkin lead current everybody wants, right? And Gary Yellen's first response on it, Gary was, that's boring. It doesn't do anything. It just sits there. Because he likes games. So it's not boring, I'm here to say. It opens and closes. This is a 5-minute trace. It actually shows multi-ion core characteristics, anomalous mole fraction. This is the Drosophila one. It's highly regulatable, so that this is the control current. This is the PMA. This is the foreign. And if we cut off the tail, it doesn't care about these things. And in fact, it was sort of interesting because, in fact, opening and closing of this channel mimics the movements we have reported that are inactivation in multiplicated potassium channels. And most recently, someone else indicated that if you knock this out or mess with it in slides, they have cardiac arrhythmia. I wish I had this one to figure that out. That just came out. So this brought us to the first functional in our hands example of the 2P weak background potassium channels, one function. At that point, many began to come out. In fact, there are 18 human genes that code proteins like this that are distributed through the body in different ways. And we began to just try and understand the nature of the beast by studying some of the individual variants. When I say studying the individual variants, I am still talking about heterologous expression, okay? You take the frog out of the child's mouth, you take out the oocyte, you use the oocyte machinery to heterologously express, and you study it. Sometimes you use tissue culture cells. In fact, we've gone to using yeast cells, or you tear the brains out of mice and you look at individual cells. That's as complicated as I get. Unless someone's free. And just to show you that I'm not a sexist pig, we put our frogs in boys and girls, although we use a different orientation. And I will say to any pediatricians in the room, these are not real because I know that frogs are parasites and that this would be childish. You would not believe the grief I've taken by having a slide like this at a pediatric meeting. One of the other examples, and I'm going to move past this very quickly, now we have 18 of these, 1, 2, 3, 4, etc. This is an example of beginning to move us into mammalian cells, a rat hippocampal cell line, and clone examples of the K2P2, they get numbered 0, 1, etc. This one also carries the name TREP, highly regulatable by phosphorylation, actually quite interesting because it moves from an open rectifier phenotype to actually being voltage dependent in any event. Each of them has their own different role and flavor. I do want to mention this variant, now moving on to the third one for just 1 second because we'll come back to something about it. First of all, I like it because it's in the heart. Second of all, it's also in the brain. It is believed to be the source of the halogen-sensitive general anesthetic response in certain cells in the brain. This is from the Bayless laboratory, and this is the pulse that I was showing you. And it's a pH-sensitive channel, so halogen makes it more active and stops firing. And then if you lower the pH, you block the channel, and then you get firing again. And in fact, what we found was that the histidine in the first 4, right after the GYG selectivity sequence that is important for finding potassium, that is responsible for it, and you block this across physiological pH ranges. And if you, I don't show you, if you mutate the histidine, then that pH sensitivity goes away. That will become relevant in a few minutes. So back to K2P1, TWIC, the channel we could not get to work. The Bayless lab showed that it was not a pseudogene, in as much as they could show transcript and protein. These are now a whole bunch of these 2P channels for the TWIC in different brain, in different parts of the brain, and it's also expressed in heart and kidney. So the question that we have then is, what's going on with this K2P1? Is it mute? Is it sleeping? What is it doing? We know that I showed you that the Drosophila one opens and closes open probability with phosphorylation. I showed you quickly one that changes its phenotype in terms of voltage dependence with phosphorylation. The pH changes the unitary conductance of the heart channel, and also it changes its number in the membrane through trafficking mediated by phosphorylation of 1433. And here we have just no function. So it is now going on 5 years that we've been studying this, and we can't get it to work. And that involves the destruction of 2 careers. No, and what would happen is pushbacks would come in, and I'd give them 1 channel that works, and 1 channel and this channel. And I'd say, if you can make it work, you're really a god. But if not, you can publish on this other channel. And indeed, Noam did beautiful work on K2P0, and then went to beautiful work on K2P2, but missing from that was K2P1, and nothing that we could do would make it work. Chimeras, deletions, point mutations, lipids, kinases, phosphatases, these two hybrid interactions find interaction proteins, drugs, whether we took them or we applied, or even the bone. The bone being a thing in Chris Miller's laboratory that when an experiment really didn't work, he would go to his office, he would pull out the bone, and he would rotate it over the sample. And then Sindhu Rajan came to the lab, and we decided to start one more time. We got 2 channels. And with Sindhu, what we said is, all right, let's start more explicitly and see if we can really figure out what's going on with this gene. So we put a GFT tag on the amino terminus and looked at oocytes, again, our expression system of choice at this point. And we said, look, there's green on the cell. It's polarized to the animal pole, but getting to the surface, and yet there is no current. We wanted to make sure, as much as we could, because this was a heterologous system, that it wasn't something that we mucked with putting the GFT, so we put a 16 amino acid sequence on the carboxy terminus recognized by a monoclonal antibody called 1V4, and found that that also went to the surface. So we believe that the channel was going to the surface in the oocytes, but it wasn't passing any current we could see. And then a miracle occurred. That's down here. You see that cartoon all the time. A point mutation, actually a single position with any residue that we put in it, brought the channel to life for the very first time. And it was acting like an open rectifier, and it was, as we expected, a GHK background current and all of that. And it wasn't that completely random in as much as it's an area that's involved in other effects on some of the other channels. So we finally had something that worked. And we stared at the sequence and tried to understand what that lysine could do. And the fact was, the best we could come up with was that it was kind of similar, not really similar, but kind of similar for a motif for stimulation. And SUMO is small ubiquitin-like modifier protein. The motif in this channel is LKKF, which is really not a classical motif, so one has to be careful about taking this seriously. It's generally hydrophobic and with a negative charge in the 4th position. So it really was not classic. But it seemed like it made sense. And to take 1 minute and talk about stimulation, the bottom line is that SUMO is a 101 amino acid protein, which is processed down to a 97 amino acid protein. It starts by getting linked to an E1 enzyme. It gets moved to an E2 enzyme. And from the E2 enzyme, it either gets directly put onto the target or there's an E3 ligating enzyme that puts it on. So it's a pathway. It's very much like the ubiquitin pathway. And where ubiquitin has many different variations of this, there's only one example, for example, of UBC9. UBC 1 through 8 are all for ligating, conjugating. Ubiquitin 9 by itself is for SUMO. So the issue is, for this to make sense, the first thing you need is the conjugating enzyme must bind to the channel. If this is real, that was the first place we started. And a yeast to a hybrid assay suggests that that's probably right, because if we took the carboxy terminus, we could get little red chicken pox on our plate if we put in UBC9 and the carboxy terminus in the channel. That argues genetically that the conjugating enzyme does bind to the channel. There's still really a problem with this, though, as we are doing the work now. And that is that, to that point in time, the 16 known substrates for stimulation were all nuclear. Transcription factors. Stimulation either sends you into the nucleus or sends you out of the nucleus. It either allows you to bind more tightly to DNA or to fall off the DNA. In a few rare cases, you stimulate, ubiquitin can't go on, and therefore you don't degrade the substrate. So The next question is, all right, UBC9 is basically being seen in nuclear pore complexes and so forth. Could it be at the plasma membrane? Would that make sense? And the answer is, first with GFP showing a uniform distribution, we could see that our overexpressed UBC9 would go to the membrane. And interestingly, when we added the channel, the UBC9 capped to the animal pole, which was suggesting that they were co-assembling and recognizing each other. And then using an antibody that made UBC9, which is interesting because it's got the antibody with the human, but the sequence of benefits UBC9 is 100% identical to human. We were able to see it in the oocyte and actually see that move when we overexpressed it. So it looks like it's in the membrane, not just in the nucleus, and it's kept co-assembled with the channel. So the next question is, is there any evidence for SUMO on the protein? And in fact, this is starting material where we immunoprecipitate the channel, overexpress from oocytes with that 1D4 tag. And this is the starting material. There are lots of proteins in a whole cell that have SUMO decoration, because this is an anti-SUMO antibody. The purified channel with the 1D4 monoclonal stains with SUMO. So the point mutation in that particular lysine of interest doesn't stain with SUMO and is smaller. And we can recapitulate that by expressing the SUMO with an encoded HA tag and showing the same thing. So now we've got the protein being modified by SUMO, which suggests that this thing may actually be real. The next component of the SUMO system is that there's a reversible component to it. We have a SUMO desumolase, which will take the SUMO off the channel. And when we looked at the protein, we could see that if we took stimulated protein, treated it with a desumolase, the molecular weight went down, and the stain for the SUMO went away. So it looks like you can put SUMO on, you can take it off. So now it comes time to trying to decide if any of this makes sense to us as channels. If you've got channels that are sitting in the oocytes that are silent, and you believe that is because they are bearing a SUMO epitope from an endogenous UBC9, then that means that these channels, which previously were silent, must wake up when we add the desumolating enzyme, or we're wrong. And in fact, when we added the desumolating enzyme, we could open the wild-type channel for the first time. And if we took the desumolating enzyme and made a point mutation in its active site, that was our control to say that just adding the protein wasn't doing anything. This was the wild type that was the simple one. So that suggests that the simulation really was opening and closing this channel. The next proof that we were opening the right channel was that this pore has a histidine just like the channel I showed you before. That suggests that it should be sensitive to external acidification, which quick is not. It's passive. So in fact, the channel we opened up with the sense is sensitive to external pH, and if we mutate the histidine of interest, then we get rid of that sensitivity. So we really believe that we were waking up the channel we thought we were waking up and could control it. The last component of is it being real is can we move this back and forth repetitively and reversibly. And Lee's plan is what I believe is illegal in some states, which we call serial patch cramp. And what he did is he started with an oocyte that was expressing both the channel and the stem. So the channel should be open. So he could go down at the single channel level and looking at the single channel and find activity. And then he pulled that patch off and the channel activity continued. And then he stuffed the patch into a cell that was wild type. So that there should be the entire simulating machinery in there and silence the channel, which it did. He then pulled the patch back out again and it stayed silent, and then he stuffed it into an oocyte that was just expressing the destimulation of channels and woke it up again. These are just single channel traces. This is a cell expressing the point mutant sense, which can't do the job. And this is a mutation of that lysine showing that it's insensitive to the whole manipulation if you do it the same way. So the conclusion at this point was that SUMO machinery is at the plasma membrane. K2P1 is regulated by simulation destimulation. That K2P1 is not switched after all these years. And the motif that we were looking for, I mean, it was clear that the SUMO motif we used was a variant, but it's present in lots of membrane proteins, which is what our lifeblood is about. So the question was, were there any other places that we should be looking? If leak channels set the resting membrane potential, and therefore are important in excitability, what about the actual mediators of excitability? Are they involved in the same pathway? That would be more interesting to Barry Yellen and more acutely interactive with excitability. So now we're thinking about voltage-gated potassium channels, and again, you guys know these. They have a sausage that moves up and down with voltage changes across the membrane, which changes the probability. And what we did was take a step back, think about where we thought this was going to happen. And we had a particular target channel of interest. And so what we did was isolate primary hippocampal neurons and put them in culture. Put hippocampal neurons in primary culture. And then use parent clams. And this is an example that sort of speaks to what we were trying to do. If you are in parent clams, you'll see some sort of fire in the wing. We then put into the electrodes, and we were going when we were breaking into the cells, 7 picomolar SUMO1, or 75 picomolar SUMO1. And what we saw is the firing rate actually began to increase. So if you're shutting down potassium channels, you might expect more activity. On the other hand, if we added the destimulase, which would activate potassium channels, lowering the resting membrane potential and silencing the neurons, that's exactly what happened. The destimulase silenced them, whereas the point destimulase didn't. And if we added a delta GG SUMO, which actually can't covalently link or keep the nature of the SUMO, nothing changed. So that suggests that SUMO might be involved in the firing of the hippocampal nerve. And the numbers that go along with this, and I hate tables, but I have to do it because it's sort of pretty in this regard, is that you can see that the resting membrane potential goes more positive with the stimulation as if there's less movement towards DK, less potassium absorbed. And if you add the step, it's as if you're opening up potassium channels, it's getting moving more towards DK. Firing rate, I talked about going up. And the input resistance goes as you would predict to show that you're opening it. So then the question is, which channels are we affecting? And in the hippocampal neurons, there are 3 primary currents of interest. There's sodium current, there's an inactivating potassium current, and there's IDR, which is a delayed rectifier. And by isolating these pharmacologically or with different pulse protocols, what we were able to see when we either looked at the control, the basal conditions, adding SUMO or adding STEM, was that the sodium current didn't appear to change. IK did not appear to change, but IDR changed dramatically. So that when the control is the box, and when we destimulate, we see more potassium flux. And when we stimulate, we see less. And in fact, this turns out to a 35 millivolt shift in the voltage dependence of activation, upon stimulation or destimulation. Well, IDR in hippocampal cells is primarily thought to be K2.1, which was our starting point for why we did this experiment, because it had a beautiful consistency between stimulation. This is just what I told you there. This is a 36 millivolt shift in the voltage dependence of IDR. So the first question we asked is, the clone K2.1, does it interact with the conjugating enzyme UBC9, and can it be stimulated? We used these 2 hybrids to show that, in fact, the C-terminus where the motif is does bind to stimulating enzymes, but the channel itself can be stimulated. And then we used FRED, both receptor bleach and donor decay, to ask whether SUMO and the channel interacted in CHO cells. And here what you're seeing is the channel with CFP, SUMO with YFP, and by receptor bleach, you're seeing the staining on the outside there. The control here is using the non-linkable SUMO. I like the donor decay because it's more reproducible and trustworthy, and it was showing a rep efficiency of 23% between the channel and SUMO is equivalent. So these two things were coming together in the themselves. And when we then went to study the clone channel in CHO cells, we were able to show the same shift of 36 millimoles in this case with the clone KB2.1. And among the available lysines, we mutated the 4 that were in anything like a consensus site, but the ones we were chasing because we thought it was right with K470, we mutated it. It's now insensitive to it. It's also insensitive when you mutated it to Q, R, E, or N. R being maintaining the charge, and maintaining the Q, maintaining the polarity, and so forth. It really has to be a lysine in that position. The other thing that we did with this was look at the rapidity of reversibility in these 5 membrane patches, and what you can see is you start at some level, you remove SUMO, you get more activity. You add SUMO, you block. You can then take the SUMO off, which you wash, and you stay in this state. And reciprocally, if you stimulate, you then wash extensively, whereas you can do this. And the issue is, I told you all sorts of enzymes are necessary to do this. This implies that the stimulating enzyme, UBP9, that we showed in the membrane is sitting there doing this linkage. The stability on the washout suggests that the destimulate is not membrane adherent because it's stable in the quiet state. The last experiment that we tried to do to see if we could connect the dots on the role of the hippocampal zone was to express the clone KB2.1 in the neuronal cells. And so the first thing we did was see that it silenced the cells because there's just so much potassium flux. But if we added SUMO at higher and higher levels, we could block it and we could get them to fire again. These lower areas are where we add PPA to block some of the channels that will fire, and we show the responsiveness to the SUMO. Whereas overexpression of these point mutants of SUMO insensitive is just stable through manipulation. It can't be blocked, whether you have more activity or less. So before the controversy came out, this is where we stood, and this is where I'm going to stop, and I have to talk about the controversy. Again, it looks like there is the stimulation machinery is with the membrane controlling the channels that we're interested in. And that it's controlling not just the background terms, but it's controlling the voltage-gated channels as well. I will thank a few people and then talk about, I saw Dr. McGoldrick, it's just the greatest idea. The most important people to mention, I have mentioned Lee Plant, who did the work on KB2.1 and much of the electrophysiology on the link channel, and Sindhu Rajan, who got that early mutation. Now, the controversy is this, that in August, he who shall not be named, so you can know it's Harry Potter, published work saying that they couldn't repeat what we did with K2B1. They couldn't get current unless they made one mutation in that lysine position. They couldn't show linkage, and therefore they were questioning the system. What we've chosen to do, because it's sort of hard to say, well, we can do it, you can't do it. You didn't ask us, here are constructs, you want to try it with this, because they use different constructs that they made differently in different cells and different systems. Was to take the most positive aspect of the paper, which is they said, look, we can't do what they did, we're using mammalian tissue culture cells, so maybe this doesn't matter. We sort of ignored the fact that they couldn't do it in oocytes either. And we moved ahead to say, maybe this doesn't translate to mammalian cells. And so we've redone the experiment in mammalian cells, which has allowed us to recapitulate everything that size patches with the leak channel the way we had done it with the voltage-gated channel. To apply breadth to the leak channel, which again shows the interaction only takes place
oops, there it is, that the interaction with the channel only takes place if the this is now, these are positive controls where we have CFP and YFP on two subunits coming together. This is SUMO interacting with high efficiency by stress with the wild type, but not interacting with E, R, or Q at that position. The delta GG, which can't covalently link, doesn't interact also, and then there's free YFP. The other thing that we've gone on to do to try and argue for the validity of what we've been studying is to show that even though the site is a variant motif of the SUMO motif, it can be modified. This is hard to do just because getting enough material from the mammalian cell still wasn't ready. We couldn't get sufficient material to do Fourier transform mass spec and then mass spec sequencing to prove it. So what we did was we used a new system developed. It's quite elegant, a group in Japan. It's a two-plasmid system in bacteria. Bacteria don't assimilate. And so it's a trisonic vector that has all the enzymes for assimilation and the SUMO. And then you put your target on another, you co-express it, you purify it, and then you have enough material. And in fact, what we found is that this non-canonical motif is found there. You can see the charge variance that you're supposed to see, and then by mass spec, you can cut it down. Simulation is really a beast to try and do by mass spec. It's hard enough to do something that has two ends when you're using mass spec. This thing has three ends to the molecule, so it makes the analysis that much difficult. We ended up mimicking the methods used for ubiquity, and part of that involves creating a SUMO that has a lysine in position 95, so that what's left behind is only a GG adduct, because what's normally left behind is so that you, again, can't use spec. So in terms of the controversy about whether this is in the membrane and whether this works, we really can't find any excuse for not seeing it. The biggest problem is that SUMO adducts are actually quite labile. And so in the transcription factor field, you will have a transcription factor that's acting as if it's 100% simulated or 100% assimilated. But when you purify the complex, only 5% of it is carrying SUMO. And so that's called the SUMO enigma. And the enigma is not completely answered at this point. It could be that on purification, the SUMO gets cut off. It could be that on, it's very clear that UBC9 stays in complex, and it could be that it holds the complex together, and then when you dissolve the cells, it all falls apart. There are some people who believe that there is memory of simulation of the protein. I do not believe in protein memory, meaning nothing is different about the protein, but it remembers that it was once simulated. And that I have some trouble with, meaning no GG, no nothing left behind. However, I do believe that it would be possible, for example, there is a truly wonderful crystal structure of 4 proteins in complex at 3 angstrom that has UBC9, SUMO attached to RAN gap, and an E3 ligase called, which is RAN-BP2, all together holding the complex, which moves from the nuclear 4 complex off to the kinetic core during mitosis. And it could be that it takes a village to hold it together, and again, when you dissolve the cell, the village falls apart and the signal comes off. So there's something going on. It is not easy, but even here we have finally been able to immunoprecipitate from mammalian cells and show the covalent complex of the SUMO on it. That was harder than in the other cells. So I can't tell you more than it works for us and it doesn't work for them, but I believe that SUMO has moved out of the nucleus to the membrane. I think it's going to be interesting and important in various of the membrane phenomena that we care about. There's a recent report at PNAS from Martins, I believe, et al. On KV1.5 showing that they see SUMO
on that protein. There's evidence for a couple other membrane proteins that's indirect but convincing. I believe that there's now work soon to come out on CFTR. So I will stop there with my dancing sumo wrestlers and answer questions that I can.
Thank you very much. The questions?
Yeah, for those of us who don't wrestle with sumo, obviously, whenever you're dealing with an intracellular mediator, you're worried about receptor modulation or cell cycle modulation or cell metabolism modulation. What evidence do you have that this is actually a target for something that's actually going to change the firing rate of these cells during some dynamic process?
So great question, and the corollary of that is if sumo is so darn important, then whatever controls sumo is going to control excitability, so what controls sumo? The simplistic answer that I have is that we started by co-expressing things waiting for 3 days and then said, look, it acts differently 3 days later. But the experiments, and I didn't say this clearly, I'm sorry I was going too quickly, for all of the both KD2.1 at the end were all in excised patches. We would pull the patch off and we'd wash by an enzyme, we'd wash by purified soap. So in that regard, we know that the modulation can be acute. So then there's the question of how acute are changes in sumo. Now you get to the extensive literature that's only about 8 years old with the whole sumo field itself. And 97 was when, 10 years, I'm getting old, keep that. RANGAP was the first simulated protein recognized, and that was in 97. We know that oxidative stress, we know that heat stress, we know that blunt instruments can change simulation status acutely in the cell of transcription factors. And it's rather fascinating because there are 4 of them. And you do something mean to the cells, and they all jump onto other proteins. They all leave where they were and jump onto other sites. But at this point, what the real master switch is, who's in charge, is really unclear. And then you can show a zillion different promoters that are repressed and others that are unrepressed and so forth. And you can show there are proteins that are now being degraded that weren't before because now the sumo is off and the ubiquitin can get onto the same site. So lots of stuff happening, but it's still really oxidative stress, heat stress. That's it. That's what's out there right now. So I don't have a good answer, and we're trying to use blunt instruments as we're trying to look at things that might relate more to channels to see if we can begin to tease out some of the mediators.
Is there any evidence that electrical activity changes the distribution or the other proteins in the distribution of the sumo? I mean, the membrane and the nucleus?
Electrical activity changes. There's in our system there is so much sumo that I'm not sure I can recognize that. When you think about trafficking in sumo, there is the fact that it will move it in and out of the nucleus to the nuclear core or not. So it wouldn't be surprising. And in that regard, thinking about the KD2.1, we know that phosphorylation state can be important for where KD2.1 goes in neurons. And we know that in transcription factors, you've got the lysine. The lysine in the motif is recognized. And downstream from that, usually in the 10 next residue, lots of negative charge is what allows that thing to be simulated. There are transcription factors that have phosphorylation sites right here. And you phosphorylate it, it gets simulated, and you dephosphorylate it, the sumo comes off. So back and forth and back and forth. Our simulation sites are close enough to one of Jim Trimmer's 18 phosphorylation sites that I do wonder how there will be, if there will be an interplay in that regard. But beyond that, I don't have more information.
You mean that actually the sumo getting off the channel is actually
modulated by, it's controlled by phosphorylation. It's not
I'm not telling you about channels now. I'm only talking about the NEF2, which is a transcription factor that is stimulated in a phosphorylation-dependent way and gets destimulated if the SUMO comes off when the phosphate comes off.
So I was going to ask, so where do you find the enzyme for destimulation?
Where do you find
Yeah.
the destimulating enzymes? Interesting as well. There are four, I believe it's four of them known at this point. They are in different subnuclear domains, except that almost all the intracellular proteases that we love and hate when we're trying to do protein biochemistry can also feed SUMO off. So it's not clear to me what has to do with an artifact for me when I try and purify proteins and what the cell is using acutely to accomplish its goals.
Have you ever seen your GFP labeled UBC lines or dynamically partitioned on and off the membrane?
We haven't done nice enough work to do that. We've seen it happen to the animal soul. We've seen threat interactions in these two hybrids, but we have not watched. And again, part of it may be that because we overexpress, you may have seen the SUMO expressed cells were yellow like crazy. And the ones that were capable of covalent linkage were lighting up the nucleus. The ones that weren't were just diffused in the cell. That's because most of the things that hold on to SUMO are in the nucleus. But we haven't watched it move. All fun things to try.
As far as the hippocampal neurons, you really think that 2.1 is the only thing that's affected, or
no, I think that there are going to be other things, but I think that lucky for us, the sodium currents, the INA and the IK weren't, and IDR being the most prominent, IKB 2.1 being the most prominent part of IDR, it just sort of stood out. But yeah, I think resting membrane potential did change. It did. Yeah, no, it
moved the records so they lined up or something.
In the raw current traces, we were lining it up so that we could line up the current excursions. In the tables that I never should put this talk, but I did, and I didn't point at today. The resting membrane potential and the input resistance all did exactly what you would expect it to do if you were opening and closing potassium channels. Again, we're abusing these cells by taking them out of their microenvironment and then putting them in industrial doses. I mean, 75 people only doesn't sound like very much, but it's a lot to a single cell. So how this relates, the elegant work I was listening to all day today about how cells really do their job, this is the first approximation of trying to understand mechanism.
Two questions. First question relates to the HMP channel. There are two lines here. How do you recognize the first one and not the second?
There are many. There I'm just going to have to depend on the literature. There are examples of stimulation sites, and it really is the one in the position next to the hydrophobic that is positioned in the Pritzker structure correctly to be modified. So I guess it's just how it fits into the active site. I thought you were going to push me on the mass spec. You might, if you know trypsin, there was an extra lysine at the end of the protein. Why is it still there? And the answer is that if you ubiquitinate or you stimulate, the enzyme can't cut right next to it. It has to go to the next available lysine or arginine, and that's why there's an extra lysine on the end of that peptide.
The second question is regarding to the hash cramming.
Yes.
How can you pull the hash even on now?
I had no. So this is where I used the line that Chris Miller taught me. He said, "Still an old man of favor." So We had no way at that time to overproduce and purify these proteins because we hadn't yet gotten that expression system going. And everything we did with the KP2.1 is now purified proteins, and we apply them. So I needed to use the oocyte as a machine to do this. And I just said, I know that Rich Kramer has crams, all right? And so I want you to serially cram. Just do an old man a favor. And the fact is, there were patches that you'd lose on the first cram, but any patch you didn't lose on the first cram, you could go through 3 different oocytes easily and not lose it. I mean, you could just, it was clearly just safe. So I think he had an end of 7 different patches in those experiments where he just went straight through and did it all for each of the wild type of the mutant and whatever. If it works, it works. Thank you very much.
Any other questions, comments? Okay, thanks. Thank you.